You're here to explore protein synthesis and the roles of proteins in the human body. And it seems you're after a valuable skill set. This topic will help you acquire a unique understanding of life's inner workings. Protein synthesis, the process of creating proteins and regulating cell characteristics, is a multi step mechanism divided into two essential stages: transcription and translation. These processes are governed by different types of RNA and crucial enzymes. Messenger RNA, mRNA, serves as the genetic blueprint for specific proteins. It carries the instructions encoding in DNA from the nucleus, where the master DNA copy resides, to the cytoplasm, where proteins are synthesized. mRNA is single stranded and contains one gene. It has a short lifespan, typically degraded soon after use. Transfer RNA, tRNA, is approximately 80 nucleotides long and forms a clover leaf structure by a complementary base pairing. One end features the ACC base sequence where an amino acid binds. In the middle loop, there is a triplet nucleotide sequence known as the anticodon. There are 64 distinct tRNA molecules, each with a unique anticodon sequence matching the 64 different codons, combinations of three base pairs of DNA. Enzymes called amino acid tRNA synthetases attach amino acids to their respective tRNA molecules. Ribosomal RNA, rRNA, forms ribosomes, the sites of mRNA translation and protein synthesis. Ribosomes comprise two subunits, small and large, and are assembled in the nucleolus of the nucleus. These ribosomes are then exported from the nucleus to the cytoplasm. rRNA is encoded by numerous genes across various chromosomes. Transcription. Transcription is the process where a copy of each gene in DNA is created to carry the genetic message from the nucleus to the cytoplasm. This copy, known as mRNA, is produced during transcription, and it is essential for protein synthesis. At A level, you will learn this sequence of events. The start of each gene on DNA is marked by a special sequence of bases called the promoter. The mRNA molecule is constructed from three nucleotides A, C, G, and U in the nucleoplasm. These ribose nucleotides attach to the DNA bases via complementary base pairing. Enzyme RNA polymerase joins the new nucleotides by forming covalent phosphodiester bonds, creating the mRNA strand. The mRNA peels away from the DNA as it is synthesized. The DNA rewinds as it leaves the polymerase enzyme. The initial mRNA, known as pre-mRNA, is an exact copy of the gene on DNA, including exons and introns. Enzymes cut out the introns, non-coding DNA sequences or segments in the mRNA molecule, joining the exons together. Exons are the coding DNA sequences segments together through splicing. Sections of the RNA transcript introns are removed this way, and the remaining sections, the exons, are stuck back together. This shortens the RNA molecule length, and the introns are degraded so that the final mRNA molecule that exits the nucleus through the nuclear pores in the cytoplasm, where it attaches to ribosomes for translation, contains only the sequence of the exon segments. Translation or protein synthesis. Translation or protein synthesis is the process where the information carried by mRNA is converted into a sequence of amino acids, forming a protein. The steps in this process include: a ribosome attaches to the mRNA molecule at a promoter region, enclosing two codons. The first tRNA molecule with the amino acid attached binds to the ribosome with its anticodon pairing to the first mRNA codon. The next amino acid tRNA attaches to the adjacent mRNA codon through complementary base pairing. Enzymes in the ribosome catalyze the cutting of the bond between the amino acid and the tRNA, forming a peptide bond between the two amino acids. The ribosome shifts one codon, allowing a new amino acid tRNA to attach. The free tRNA leaves to collect another amino acid and the cycle repeats. The polypeptide chain elongates one amino acid at a time, peeling away from the ribosome and folding into the protein. This continues until a stop codon is reached, causing the ribosome to disassemble, releasing the finished protein. This video is your ticket to grasping the knowledge of how your body builds and uses proteins. Protein synthesis, starting with transcription and ending with translation, is the human factory assembly line. It's a step by step process where DNA's blueprint is turned into functional proteins that provide the tools the body needs to survive. From DNA to proteins, each step is a piece of the puzzle that brings you one step closer to understanding the big picture.